My laboratory is broadly interested in how macromolecules adopt tertiary structures to impact their function in cells. And my lab is interested in the regulation of protein synthesis by the ribosome, which is the most conserved macromolecular complex in any living cell, and also it is makes all proteins in all living cells. My name is Dunham. I'm an associate professor in the Department of Biochemistry in the Emory School of Medicine, and I'm an associated faculty member in the Department of Chemistry. As an undergraduate, I was always interested in science classes, and really got my first experience doing organic synthesis at Albany Medical School, where I was synthesizing small molecules to potentially inhibit cancer. And I realized straight away that undergraduate research was something that I wanted to continue with. And so the next summer I worked at the University of Texas Galveston Medical Branch, where I studied how different repair enzymes interacted with DNA using biophysical techniques. And I got to read quite a lot about nucleic acid protein interactions, and went back to my senior year at Barnard College, which is part of Columbia University, and with a renewed interest in studying RNA molecules, which I had thought were very, very important and interesting, mainly because the Nobel Prize had been awarded for the discovery of RNA enzymes. And so the idea that RNAs could do enzymatic reactions like proteins was a very intriguing question to me as an undergraduate. So as a graduate student, I was really interested in studying the structure and function of RNA enzymes, in particular using X-ray crystallography. So I joined Bill Scott's lab at UC Santa Cruz, and I used time-resolved crystallography to study the hammerhead ribozyme, which is involved in the viral replication cycle. I then next, for my postdoc, wanted to study a much larger macromolecular machine using also X-ray crystallography, and so I joined Ram Christian's lab at the MRC Laboratory of Molecular Biology in Cambridge in England. And there, we were really trying to push the boundaries of what we could do using X-ray crystallography, and I joined a great team there where we spent many days and weeks and nights going to the synchrotrons across Europe to collect data on our sometimes good and sometimes bad diffracting crystals. And so I felt like we were pushing the boundaries of what we could do by solving the structure of the ribosome, and it was really the most invigorating scientific time of my life. I then decided, after about 5 years as a postdoc, to look for positions back in the US, and that's where I found a position at Emory University in the Department of Biochemistry. I started my own laboratory in 2008, and I was very interested in extending some of the work that I had done as a postdoctoral fellow in Benke's lab, which is understanding how protein synthesis is regulated. At that time, the main technique we were using was X-ray crystallography, but that soon morphed into other types of structural biology and also biochemical techniques to understand the overall molecular mechanism of how protein factors, different RNAs, different environmental conditions can regulate translation. I'd say half of my lab now is really centered on understanding how toxin antitoxin systems control translation. And what's really great when you're able to run your own lab is just seeing where the research takes you. So if you had asked me when I first started my lab what I would be working on, it'd be completely different from what actually we work on today. And it's down to the fact that your interest changes once you get some exciting data. And I would view the toxin antitoxin field something really interesting that we got into just haphazardly, and it's really turned the direction of my lab into a totally different new direction. I get asked this a number of times, so what's the definition of a biochemist today in our scientific world? And I think it's a complex question because I think biochemists can contribute to the scientific endeavor in many different ways. So I think as biochemists, we're all interested in understanding how things work, how biology works, what is the mechanistic or molecular basis of biochemical processes, and usually that involves biophysical approaches to understand binding, catalysis, but also incorporated with structural biology approaches, including NMR, X-ray crystallography, and single particle cryo-EM. And the idea is that you bring in all the techniques together to be able to give a very well-rounded and mechanistic view of the scientific or biological process that you're studying. I think it's really important to have quantitative approaches in biology today, and I think that's why biochemists are really important to the scientific community. As an undergraduate, I benefited greatly from the generosity of a number of scientists that I worked with, including, of course, professors, but also the postdocs and the graduate students in those labs. And so I think mentoring is really important at all levels of science, and I know that I wouldn't be here today if I didn't have incredibly good mentors. So that is a defining goal of my lab, is to increase participation from a number of diverse communities here at Emory, and I think it's really important that you get different perspectives in science. Science is international, and we, in addition to international, we need diverse voices, including gender and race. And so I tried to provide similar experiences like that in my lab. And the undergraduates that work in my lab have real projects. We want to give them a project where they can own it, but also experience the highs and the lows of the project, because that's what all scientists undergo when they embark on a scientific project. So in order to give these students real experiences, you also have to provide good mentoring. And it's something that I pride myself on, that the graduate students and postdocs in my lab take seriously and mentor our undergraduates. So the ribosome is a target for about half of all clinically relevant antibiotics. So clearly it's an important molecule to understand on a molecular basis. The Nobel Prize that was awarded in 2009 in chemistry was for the discovery and identification of how antibiotics interact with the ribosome, and that was awarded to Tom Stites, Ada Yonath, and Venki Ramakrishnan, my postdoctoral advisor. It was a surprise for us to hear that he had won the Nobel Prize. At that point, I had been at Emory University for about a year since leaving his lab. So even though it was a surprise, it was the culmination of just many years of working on sort of the structure of the ribosome that came together, and I think it was a great honor. There were a number of people in the field that had worked for decades on the ribosome, and so from that perspective, it was hard to actually pinpoint 3 people who could win it, but I do think that they did select the researchers that had the biggest impact on understanding how antibiotics interact and inhibit the bacterial ribosome. It was also a turning point in how we think about antibiotics and how we could possibly even engineer different types of antibiotics to interact with the ribosome. So from that standpoint, biomedically, it was incredibly important to solve high-resolution crystal structures of the ribosome. From a fundamental point of view, the ribosome is also incredibly interesting because it's the largest asymmetric RNA protein macromolecular machine in your body and all cells. And we can learn a lot about principles of assembly of macromolecular machines, disassembly, regulation, and also dysregulation, which we know is important for a number of different disease states, or causes a number of disease states. And so I think this whole race towards understanding the ribosome in atomic detail was incredibly exciting to be part of. And actually there's a new book by Venki that just came out that discusses his race for understanding the ribosome in molecular detail. That's called The Gene Machine, I think. And I've heard it's quite a thrilling book about sort of the trials and tribulations that one undergoes in trying to solve a very important but also very challenging biological problem. And I think it's a great book for all undergraduates to read, to get an understanding that science isn't always successful and there's always going to be challenges, but how you overcome those challenges is almost as important as the answer to your question.